Welcome, welcome. We are getting ready to start chapter 9 videos. We're going to do something a little bit different in chapter 9. We are going to not cover all the chapter. We're only going to cover 3 sections, and we're going to do them slightly out of order. Okay, so we're going to cover 9.3 first, then we'll jump back to 9.1, and then finish off chapter 9 with 9.5. So let's get started. We have a few objectives with chapter 9. Okay, chapter 9 is all about bacterial genomes and evolution. Okay, so what causes changes in these bacterial species? Okay, so you're going to be able to describe how genes move between genomes by the processes of transformation, transduction, conjugation, and transposition. This is all what we discussed in 9.1. You'll be able to explain the process of natural selection and how it applies to bacteria. And then finally, we're going to be able to explain microbial taxonomy and how we identify different microbes. Okay, before we start into chapter 9, let's do a quick review of chapter 8. So in chapter 8, we talked about mutations and how they happen and the different types and how they affect proteins. Remember, a mutation is a change in the DNA sequence. Okay, so swapping out a base or a couple bases for one or deleting some bases. These mutations are the source of genetic variability in organisms. Okay, this is going to account for the differences between species. In fact, most life on Earth is diverse because of this process of evolution and this process of going through mutations. Mutations are going to cause a species to diverge from each other. So we're going to get 2 separate species. Okay, but the thing that we really don't talk about as much as we should in biology is that a lot of these mutations that happen at the DNA level are very random and they are very slow to happen. Okay, it takes a lot of generations in order for these things to occur. So they occur at a very low rate. Okay, so chapter 9.3 is all about natural selection. Okay, so one of the things that you're going to have to be able to do when we come out of 9.3 is you need to be able to explain how fitness is a trait that depends on the environment. Okay, and then you're going to have to take that process of natural selection and be able to apply it to environment-dependent fitness when it comes to the evolution of bacteria thanks to antibiotics. Okay, so natural selection, if you remember back to biology, hopefully in your biology class they talked about this. So natural selection is the process of mutations allowing variability in a population. Okay, so think back, these are our pepper moth example. So the ones with the black wings and the ones with the white wings. Okay, so fitness, so how easily an organism can adapt to its environment is fitness, okay? So fitness is going to affect how that organism survives and whether it has reproductive success. Now, there's one main goal of all living things, and that's to have reproductive success. This means that they want to survive long enough to reproduce and pass their genes on to their offspring, to the next generation. Okay, so certain mutations, certain characteristics are going to allow one organism to survive over the other. Now, if you remember back to our pepper moth example from biology, before the Industrial Revolution, a lot of the trees that the pepper moths lived on were white and black. So the ones that had the white wings with the black speckles, they blended in, so they wouldn't be eaten by birds and other animals. But the pepper moths with the black wings, they didn't blend in, they stood out. Okay, so they were one of the first ones eaten. So when the Industrial Revolution happened, most of the pepper moth population had white wings and just a very few with black wings. But once we got factories up and running, started to mass produce things, they're burning coal, and that coal added ash and soot to the surface of those trees. So the surface of the trees weren't white anymore, they were black. Okay, so we had a complete reversal in the environment. Okay, so the black pepper moths now were blending into the trees while the white ones were not. So we saw this shift in the genetics of this population where now most of the surviving pepper moths had the black wings and very few had the white wings. Okay, so this is an example of fitness where your genes are going to allow you to survive and reproduce better. Okay, when this happens, so when that change came with the Industrial Revolution, the environmental effect of the Industrial Revolution selected for the pepper moth with the black wings. Okay, they were able to survive and reproduce better. Okay, so in bacteria, they're going to have variants or strains that are going to be able to do the same thing. When they're selected for, they're going to be the ones that survive and reproduce. Okay, the thing is, they're going to have specific traits that allow them to do this. Okay, so one of the traits we talked about with the pepper moths is the wing color. So for bacteria, it might be the ability to break down lactose. It might be the ability to live in an aerobic environment. Those are both traits that allow them to survive in different environments. Okay, so when we select for certain variants or certain strains, when there's something like an environmental change, maybe the ability of lactose is going to allow certain organisms to survive, certain strains of bacteria to survive. Or maybe all of a sudden our anaerobic environment becomes aerobic. Okay, well, if we have the enzyme to allow us to deal with those reactive oxygen species, we're going to be the ones that survive, and the ones that don't are going to be the ones that die out. Okay, so this whole idea of selection, it's not too different from the selection that we do when we talked about what our plates, where we only allow certain organisms to grow. This is pretty much the same thing, except this is how it naturally happens. This is what happens in an environment. Okay, most natural selection happens because of an environmental change. Okay, so over many generations, as we select for certain traits, the descendants are going to have different traits than their ancestors that are going to allow them to be better suited, but more fit for the environment. Well, and if they can't adapt to these environmental changes, they're going to go extinct, right? This is kind of what happened with the dinosaurs. They had some environmental event that happened and they weren't able to survive it. So now they are extinct. Okay, so either we adapt to our new environments or we die. Okay, so let's look at an example. This is very similar to the pepper moth example I was talking about on the previous slide. Okay, so we have this population of mice. Okay, so this population of mice is normally found in a wheat field. Okay, so in this population, there are more mice with the tan fur color than with the darker fur color. Okay, as we can see here in this first example, right? We have less with dark fur, and we have more with tan fur. Now, there's going to be an environmental change that happens. So there's a tsunami that has swept in and has washed these mice from their natural environment of the wheat field, and they're now in a rocky environment. Okay, so previously these mice with tan fur, they were able to blend in. They're not going to be the ones that get eaten by the hawks because they can better camouflage themselves in their environment. Well, now that they have had this environmental change, they're now in this rocky landscape. They're really sticking out, right? They're going to be the ones that are easily seen, and the hawk's going to swoop in, and they're going to be the ones that are eaten. While now the ones that have the darker colored fur, that were previously the ones that were being eaten first, now they're camouflaged great. They're blending in. Okay, they have the genes that are going to allow them to survive in this new environment. Okay, so they're going to be the ones that are going to be able to reproduce, right? All our little other tan guys have been eaten by hawks. Okay, so now what we're going to see after a few generations is that our mouse population is now going to be mostly dark colored. So they're going to have the dark fur color, and very few are going to have the tan fur color, which is the complete opposite of what they had before that environmental change. This is what natural selection does. Okay, so natural selection, we want to survive. We want to be the fittest for the environment so we can reproduce and pass our genes on to the next generation. Okay, all living organisms follow this process. Okay, so one of the really interesting things about bacteria is because they have such quick generation times, we can actually see evolution in action. We can see what happens when there's an environmental change. Okay, so the forces that drive selection, this natural selection process, is something that we call a selective pressure. Okay, so for the mice in the previous example, the selective pressure was that tsunami wave that washed them into that rocky environment. Okay, for the pepper moths in the first of the example, the selective pressure was the Industrial Revolution and the soot and smoke and ash that was produced by these factories. Okay, so environmental conditions are going to be what are going to put these selective pressure on these bacteria. This could be things like a change in temperature. a change in nutrient availability, or in our case, the presence of antibiotics. Antibiotics are a very great selective pressure in a bacteria community. Okay, so these selective pressures are always going to select for the ones that are best able to survive and reproduce in this new environment. Okay, and those that are fittest, their genes are going to become the dominant ones in the population. So let's look at how this works with bacteria. Now, there's a lot of people that would argue, this is more like a philosophical debate, that our use of antibiotics is not natural. This wouldn't normally happen, and so it's more into artificial selection, but we'll leave that for other people to discuss. Okay, so we have this population of bacteria. Okay, so most of them are these yellow guys. Okay, they are the non-resistant, so they don't have any antibiotic resistant genes. While these little red guys here, they're the ones with the antibiotic resistance. Okay, so they get into a host, they cause an infection. Okay, their host is going to go to the doctor and say, I'm not feeling great. They're going to identify that as a bacterial infection. And what's going to happen is they're going to be prescribed antibiotics. The host is going to take the antibiotics. And what's going to happen is all of these yellow guys who are susceptible, not resistant, are going to succumb to the antibiotics. So the antibiotics are the selective pressure here. Okay, so after about a day or so of taking antibiotics, the host is going to have a few bacteria left. Okay, so most of the yellow guys are gone, but our red guys, our resistant ones, are still there. Okay, so we get done with this antibiotics course. Well, what's going to happen now? Most of the yellow guys are gone, but we still have those red guys. They were the ones that were selected for in this scenario. So as generations go on, we're going to see more and more of these red guys and less and less of these yellow guys, until finally a few generations later, most of those bacteria in that strain are going to be antibiotic resistant now. Okay, this is an example of antibiotics as a selective pressure. Okay, so let's talk about case history. Let's see this in action. So our case history is called the bacteria that kept coming back. Okay, so we're going to set the stage. This case history is about Harrison. Harrison is a 73-year-old Asian man with kidney failure. Okay, so he's been admitted to the hospital. He has a high fever and hypotension, which are both very classic symptoms of bacteremia, which means that he has a blood infection. Okay, so since he came in with those symptoms, they're going to take a blood sample. Okay, so they take a sample from Harrison, they took it to the lab. So because it's bacteremia and it's in the blood, they're going to grow on special blood agar. In this case, they're going to use a horse blood agar to grow the colonies. So what they find is these white guys. So they take these and they grow them, and they use a bunch of different techniques, and they confirm that Harrison has MRSA, so methicillin-resistant Staphylococcus aureus. Okay, so yes, he has a blood infection, and it happens to be one of these antibiotic-resistant ones. Okay, so where he's older and he's coming in with MRSA, we have to figure out what the source of this infection is. So investigations in his home uncovered the fact that his dialysis line wasn't properly flushed and properly cleaned, and that's the source of his infection. That's where he got the MRSA. Okay, so with his strain of MRSA, it's still susceptible to vancomycin, which is a very, very strong antibiotic. It's an antibiotic that's going to target the cell wall synthesis. Okay, so he's treated with IV vancomycin, but it's not working as well as it should. So the bacteremia, his infection, is persisting. Okay, so they took another sample from Harrison, and they tested them for whether they're susceptible or resistant to different antibiotics. Okay, so unsurprisingly, they found that these bacteria were partially resistant to vancomycin. Okay, but they were sensitive to rifampin, which is going to target RNA polymerase. They are going to be sensitive to ciprofloxacin, which is going to target denigyrase. and linazolid, which is going to target protein synthesis. Okay, so they're going to administer a cocktail of these different antibiotics to Harrison to see if they can get this infection underneath control. Okay, so they're going to give him this antibiotic cocktail. He's going to be on it for 3 weeks. His symptoms are going to improve, but within 3 weeks after going off the antibiotics, his infection returned. Okay, so they pull another blood sample, and they now show that his infection is now resistant to both rifampin and ciprofloxacin. Okay, so that means that the only one that they're still sensitive to is linazolid. Okay, so they're going to give him IV linazolid. So they're going to administer it alone through IV for 6 weeks. Okay, so he goes through this 6-week course. His symptoms have improved, but as soon as they take him off the antibiotic 5 days later, his infection has returned and he's back in the hospital. Okay, so they are going to prescribe another 6-week linazolid course. Okay, but the thing is, when they take a new blood culture after this 6 weeks, well, they're going to see something really interesting. This new blood culture, when they grow it and compare it to that first one that they took, the colonies have changed. They're not the same size. Okay, this is never a good sign when you have a distinctly, easily distinguishable physical change that has happened. Okay, so these are Harrison's original colonies. Okay, so we see that they're white colored, a little bit cream colored. They're a decent size. They're not overly big. Okay, but these are his new colonies. See that size difference? They're still about the same color, but the size difference is going to tell us that something has changed. Okay, so we're going to call this the small colony variant. When they take this new variant and these colonies and test them for antibiotic resistance, some heartbreaking news comes back for Harrison. Okay, these small colonies are partially resistant to linazolid, which is why it hasn't been working as a treatment. Okay, and not only that, they are fully resistant to every other antibiotic we've tried. So the rifampin, the ciprofloxacin, the vancomycin, they're now fully resistant to those. Okay, so because this new variant is not responding to linazolid the way it should, they're going to switch to Harrison's treatment. Okay, so they're going to switch it to trimethoprim methoxazole. Okay, we're just gonna call it trimethoprim. Okay, so because of his infection, this new antibiotic treatment has to be administered indefinitely. Harrison's gonna be on this antibiotic for the rest of his life. Okay, unfortunately, even though he was on this course of antibiotics, it didn't respond well to them. It didn't work. Okay, they couldn't fully eliminate this infection from Harrison's bloodstream. Okay, so eventually Harrison dies from this blood infection. Okay, now Harrison had some things that are stacked against him, right? He was older. He's an elderly patient. He was on kidney dialysis, so his immune system is weakened and not up to par. Okay, so combine that with the fact that they were antibiotic resistant, Harrison had very small chances that he would be able to fight this infection. And we can see that all these things worked against him, and Harrison ultimately passed away. He couldn't, the treatment didn't work. Okay, so let's talk about this. Let's talk about why evolution is so important that we're teaching it in microbiology. Why do healthcare professionals need to know about evolution? Why is it critical? Okay, one of the things that healthcare professionals have to understand is that it's very important to ensure that their patients take a full course of the antibiotics as they are prescribed. Okay, so if your antibiotics say that you have to take 7 days worth of antibiotics, you need to take those 7 days, and you need to take the amount of pills at the certain times that you are supposed to. If it's 10 days, you have to take them for the full 10 days. Okay, because taking antibiotics as prescribed are going to prevent the spread of these antibiotic resistant strains. Okay, so let's talk about what happens here. Talk about what happens when the patient does what they're supposed to. Okay, so let's start with our previous example. We have our bacteria that are these yellow guys that are non-resistant, and we have the resistant guys here. Okay, so you're going to get your antibiotics, you're going to start to take them. After the first day, we're going to significantly decrease our bacteria population that is causing our symptoms. Okay, so we're going to have a few of these yellow guys left, and we're going to have these red guys left. Okay, so what's going to happen is, even though most of the bacteria are killed off in those first few days, these antibiotics are important because what they're going to do is they're going to keep the population of bacteria low for long enough for your immune system to ramp up and take care of the rest of the infection that the antibiotics can't touch. Okay, so it's going to keep it low so our immune system is starting to pick up. Okay, we see a problem. It's starting to ramp up. And then finally, it's going to reach the level it needs to in this infection cycle that it's going to be able to take care of that rest of the infection, even though the antibiotics can't kill these guys. Okay, this is why it's important to take your antibiotics as prescribed, because even though it's not killing those resistant ones, it's keeping the population of bacteria low enough that your immune system can come in and take care of what the antibiotics can't. So this is going to clear the infection. Let's talk about what happens when your patient doesn't do as they are told. Okay, this happens all the time in the healthcare field. It's actually one of the reasons why I decided not to go into the healthcare field. I have a problem with people not listening to me when it's good for them. So what's going to happen if they don't take their prescribed antibiotics? Okay, so we're going to start with the same example, right? We're going to start with our resistant and non-resistant guys, where the non-resistant ones are going to be the majority of the bacterial population in this infection that's causing these symptoms. Okay, so the patient goes in, they get their antibiotics. Okay, so they take them for the first 2 days and they're feeling better. Like, their symptoms are going away. Why do they want to keep taking the pills? Okay, they're feeling better. It's gone now. Okay, so they stop taking their pills after the first couple days. Well, most of our antibiotic susceptible population has been killed off by the antibiotics. Okay, leaving just a few of those guys and our resistant guys. Well, they're no longer taking the antibiotics, so it's no longer suppressing the bacterial population. So the bacterial populations are going to be able to reproduce. They're going to be able to go through binary fission. So what we're going to see with every generation that goes by, we're going to get more and more of these resistant bacteria. Okay, well, at some point, we're going to have a large population of these resistant bacteria. The infection's back, the symptoms are back, the patient's not feeling good, except now they go to get antibiotics and the antibiotics aren't going to work because this bacteria has now been selected for, so it's now resistant to that antibiotic. Okay, and we're going to have a hard time getting this infection treated. Okay, so let's talk about this. Not only should we make sure that our patients are taking the prescribed antibiotics course, okay, doing the full time, but we also need to be careful about the overprescription of antibiotics. Okay, so one of the things that we know when our first antibiotic was penicillin, right, the one that grew from the mold, well, we don't really hear about penicillin being prescribed anymore. There's a reason for this. So when penicillin was first discovered and they saw how effective it was, they prescribed it for everything. Okay, so we started to, because they were prescribing it for so many things that it didn't need to be prescribed for. Oh, you have the sniffles. Here's penicillin. Oh, you had the flu. Here's penicillin. Okay, so what happened is with this overprescription of penicillin, well, we did what happened in the previous example, right? We have a few strains that were resistant penicillin. Well, now we've killed off all the ones that were susceptible for it, leaving all the ones that are resistant. So there's a lot of bacteria strains that don't respond to penicillin anymore, and this is why. Okay, so not only should we watch out for the overprescription of antibiotics, as well as making sure patients take their full prescribed course. Okay, so this is a interesting curve. Okay, so this is what's happening in terms of pain with the symptoms in your patient. Okay, so the line is the bacterial infection. Okay, so we have symptoms, right? Our patient is in pain. Okay, they come and finally get help. We prescribe antibiotics. Okay, they start taking the antibiotics. Well, their bacterial infection is going to decrease. Their pain is going away. They're going to start feeling better. Well, if they continue with their course of antibiotics, the bacteria population is going to keep decreasing, and then finally we're going to reach a point where the infection level is taken care of. There's no more bacteria, there's no more symptoms. They have finished their course. Okay, so the same thing's going to happen for those that stop their course early. Okay, so they've come in, they're in pain, we've prescribed the antibiotics, the bacterial population is going away. Okay, they're feeling better, there's no pain. So they're going to stop taking their antibiotics right here. Now, this curve keeps going down because they still have antibiotics in their system, right? It just doesn't go away as soon as you stop taking them. Okay, so we're going to see a decrease in the bacterial population till we reach the point where there's no more antibiotics in your body. Now, when they reach this point, there's nothing suppressing that bacteria population, so they can go on and divide again, and the bacteria population is going to increase, increase, increase. The infection's going to come back. They're going to start to feel terrible again. They're going to start to feel pain. Well, they're going to reach that point where they went in to the doctor the first time, where they're in so much pain, the symptoms are so bad that they need help. Okay, but the thing is at this point, we don't know if the antibiotics are going to work anymore, or if we now have a resistant strain. Okay, this is why it's critical that healthcare professionals have a good understanding of evolution, because this is going to drive what we prescribe to our patients. Okay, so that is 9.3. We have talked about natural selection and selective pressures and how antibiotics can lead to the rapid evolution of bacteria by using them as a selective pressure. Okay, so we will see you again for the video for 9.1.